Good day, everyone. My name is Albert Doughan. I'm a master's student at Nkrumah University of Science and Technology in Ghana, and I am here to present on my current master's thesis on unraveling the genetic signatures of African Burkitt lymphoma patients. All right, so I'll delve right into it. Okay, so Burkitt lymphoma is a childhood cancer that mostly affects the lymphatic system. This includes the lymph nodes, the white blood cells, and other cells of the body. It usually affects the B cells, as I said earlier, and sometimes we call it the B cell lymphoma. It mostly occurs in children and sometimes in adults. And um, the ratio of boys to girls is about 4:1, which means that the disease is very prevalent in males than in females. Okay, so a little bit of genetics. Burkitt's lymphoma involves two chromosomes, that's chromosome 8 and chromosome 14, or the Q arms. So this image here shows the chromosomes of a normal child. So the CMYC gene sits on the long arm, yeah, that's the Q arm, and immunoglobulin H gene also sits on the long arm of chromosome 14. So in Burkitt's lymphoma, the CMYC gene moves and it sits right below uh, immuno- immunoglobulin H gene, and every time this gene is expressed, the CMYC gene is also expressed, and it leads to Burkitt's lymphoma. Okay, so there are three types of Burkitt's lymphoma. The first one is endemic Burkitt's lymphoma, which is very common in Africa, Asia, and South America. This usually found in places where malaria is very endemic and common. Yeah, so Ghana is one typical example of that. There is also sporadic Burkitt's lymphoma, which is very common in Europe, and there is also the third one, immunodeficiency Burkitt's lymphoma, and this is very common in HIV patients. The focus of this talk will be on the first one, endemic Burkitt's lymphoma, which is highly prevalent in Africa. Okay, so some few symptoms of Burkitt's lymphoma. There's a swelling around the facial region. I'll show an image in the next few slides. Around the jaw and the cheek, it usually swells and becomes very big. And children with this find it very difficult to breathe. Sometimes the swelling can be really big, and yeah, difficulties in breathing is very common. Uh, in diagnosis, the main method used is biopsies, and there is um, a starry sky appearance in the biopsies. I'll show an image in the next few slides. Yeah, so this is a child with Burkitt's lymphoma. So as we can all see, there's a swelling around the cheek area, and obviously, such a child will find it very difficult to breathe. There is also same thing. The cheek is swelling. There are others to the cheeks don't swell. Instead, the stomach region, yeah, it swells a lot, and it's also really bad. And there's the starry sky uh, appearance of Burkitt lymphoma patients, and yeah, this is used to diagnose this condition. Okay, so my main goal was to identify differential expressed genes between Burkitt lymphoma patients and non-Burkitt lymphoma individuals using a pool of data from Africa. And my other aim was also to find the biological pathways the genes are implicated in, and this will serve as a guide for drug designers to develop something to cure Burkitt lymphoma. I also use data from this work to benchmark tools that are used for differential expression analysis. Okay, so for the data set, I used 100 samples. 50 of them were cases, and the other 50 were control. I got the data from Lombardo et al. And Abate et al. The data were found from NCBI, SRE, and uh, DNA Data Bank of Japan. Yes, so that's where I got the data from. The normal samples were also gotten from the 1000 genome projects in Africa. Okay, so the data were downloaded, then checked the quality of the reads, and then the reads, that were bad were trimmed off. The sequences were then aligned to the human reference, the transcriptome using a Callisto aligner. Okay, and uh, the alignment stats were they were collected using feature counts, and then differential expression analysis was performed using the SEC2. Finally, gene enrichment analysis was also done using G-Profiler and David annotation tools. So this shows the results for the first part of the work where I trimmed. So before I checked the quality of the reads using FastQC, and I found that well, some of the reads were good, others were bad. Yeah, the bad ones were quite a lot of them, so I had to trim. So after trimming with Traumatic, I got this, and this looked good for further downstream work. Then I went ahead to align, and after alignments, I got this figure, and from here you can see that majority of the reads passed since they were more than 80% alignment. The literature says that if alignment rate is more than 75%, you're good to go. So 75% will be somewhere here, and almost all the reads fall beyond that, which is a good thing. Unfortunately, there is one sample that didn't pass. I singled it out, I trimmed it several times, it still didn't work. I changed the tool for trimming, it still didn't work. So I'm still trying to find a way around these samples, this particular sample. So before going ahead to find differentially expressed genes, we did some exploratory data analysis, which included PCA and density plots. Now, this provided information about patch effects and other parameters we are supposed to assess in our data sets. So this is a density plot before and after normalization. The normalization we used log2 and VST. Okay, so from these plots, they are quite uneven, and this suggests that there's a presence of batch effects in our data, which will be dealt with in subsequent steps. And also, since the distribution of all the plots are similar, it shows that there are no uh, outliers in our data set. Okay, so this also the PCA plots before and after normalization with log2 and VSC. So before normalization, we found that there are some misclassifications concerning the disease and the normal cases. After normalization, we found that the normal samples grouped very well, and the disease samples also uh, they clustered together. Yes, so we know that our data set is now okay for further downstream work. So, as I mentioned earlier, the density plots depicted the presence of hidden batch effects, and here was corrected using the surrogate variable analysis approach, where two sets of surrogate variables were estimated and added to the design of the data. So from this chart, it is evidence that the batch effects are more profound in disease samples than in the normal samples. Yeah, so we can see that from the disease group, they are more spread compared to the normal sample in both in both the first set of SVA and the second set of SVA. We then moved on to plots, uh, MA plots, and this shows the genes that are down-regulated and those that are up-regulated, and those that are neither up or down regulated. They are in the ash ash color below, and those above zero are up regulated, and those below zero are down regulated. Okay, so this bar chart shows uh, the percentages of genes that are down regulated, those that were of low counts, and those that were not either up regulated or down regulated. There were no um, outliers in our data, so this is zero, and those that are up regulated. And we found that those that were down regulated constituted the maximum number, about 32, yeah, 32%, and those that were upregulated was around 22% of all. Okay, so we were able to find 7,074 differentially expressed genes between the normal samples and uh, the disease samples. We then went ahead to find the top three most significant genes, and we found them to be BMP7CCL22 and BACH2. And all these genes are involved in gingival growth, arthralgia, and collagen formation. Gingiva is somewhere located around the jaw area, and it shows that it's implicated in Burkitt lymphoma pathogenesis. Okay, so that will be the end of my presentation. So in future, we would benchmark tools that are used for differential expression analysis. We found that there are quite a number of tools that can be used for this work, and uh, researchers find it quite daunting to choose which tool to use in their work. So I plan on adding this part to the work, and I'll also include data from Asia and South America, specifically Brazil, and surprisingly, the whole of Ghana, there's no work that has sequenced the genome of Burkitt lymphoma patients, and I also intend to do that in the future. Okay, I would like to thank Ken University, where I am right now. I would like to thank uh, Fogasi for the funding through Waslejbu projects. I would also like to thank Dr. Pandam Salifu, my supervisor, and finally, I would like to say a big thank you to all my course mates and friends who made this possible for me. Okay, I'll be here to answer your questions, if any, and I also accept any suggestions in order to make this work really good. Thank you for your time.